right, it is now 2 p.m. here in Indiana. It is 7 p.m. in London and 4 a.m. in Tokyo, if anybody's joining us from over there. This is going to be the Publishers Panel, again, with our two guests. My name is Andy, and I have with me today Dr. Rita Strack. Rita is at Springer Nature, a senior editor. Rita obtained her PhD in biochemistry and molecular biology from the University of Chicago. While there, she worked with Benjamin Glick and Robert Keenan to engineer improved variants of the red fluorescent protein DS-red, and also studied the chemical mechanism of chromophore formation in DS-red. She continued her research as a postdoctoral fellow in Sammy Jeffries' laboratory at Weill Cornell Medical College, where she developed fluorescent reporters for live cell imaging of RNA, such as Spinach2. It's a cool name. She handles imaging, microscopy, and probes, along with protein and RNA biochemistry content for the journal. Rita joined Nature Methods in November 2014. Rita, thank you so much for coming on today. It's great to have you.
Thank you. You're welcome. Can you hear me?
Yes, we can hear you. Wonderful. And then our second panelist is Dr. Andrea Scharnhorst. Dr. Andrea Scharnhorst is a senior policy advisor at DNAS. In her academic career, she moved from statistical physics to philosophy of science, scientometrics, and information sciences. She has published on models of innovation and science dynamics, the Matthew effect in science, the evolution of classification systems, the use of digital research archives, visual interfaces for information navigation, and the application of linked data in the humanities. Often her work entails the transfer of concepts and methods at an interface between physics and social sciences and humanities. Dr. Scharnhorst coordinated and participated in several European Union and national funded projects. From 2013 to 2017, she was chair of the Cost Action TD 1210 No Escape. And most recently, she was one of the PIs of the Digging into Data project, Digging into the Knowledge Graph. It is wonderful to have you on. I would now like to give it over to Rita.
Okay, great. So Andy, thank you so much for inviting me to participate in this cool event. And nice to see you in person, virtually. So today I'm going to spend about 20 minutes talking about what is a multi-scale human. And just basically from the perspective of a journal editor, specifically one at Nature Methods. But just quickly, I'm going to introduce the Nature portfolio journals and Nature Methods in particular. I'm going to talk to you about sort of the history of spatial biology at Nature Methods and sort of that we're excited about it, why we're excited about it. And then Andy gave us a list of questions he thought would be of interest to this session in particular. So I'm going to go through several of those. So that's like publishing challenges and opportunities in this space. So just a quick introduction to the Nature portfolio. So we are owned by Springer Nature, and we have a lot of different journals in our brand. Perhaps many of you have heard of Nature. That's our flagship journal. Papers that are published in Nature should be of exceptionally broad interest to all scientists. Then we have the Nature research journals, of which Nature Methods is one. And so we tend to think of these as of interest to every researcher in a specific field. So for example, Nature Neuroscience, we think it should be read very broadly by all neuroscientists. Nature Methods is special. I think our audience is a broad audience of biologists. And we tend to publish work by developers, but for a broad readership and a broad user base. Nature Communications is our flagship open access journal. They publish highly significant advances that influence a field. They don't have to have exceptionally broad appeal, but they have, of course, excellent science. Then we have the communications journals that are growing. These, again, publish very high quality work, but maybe the audience is a very particular field. So for example, in microscopy, let's say, instead of being of interest to maybe all cell biologists, maybe it's of interest to a subset of people working in a subset of a discipline of super resolution microscopy, for example. So very high quality work, very interesting work, but the audience is perhaps smaller. And then Scientific Reports is our sound science journal. We don't have to have a high potential impact or something to be very, very innovative. It just has to have sound science and have an important new conclusion. So that's the Nature portfolio. I'll tell you guys a little bit more about Nature Methods. So we just had our 20-year anniversary in October. We are a team of 7 editors with diverse research expertise. We are all professional editors, so we all have a PhD, but this is my full-time job. I'm not also a PI anywhere. We are a high visibility forum for publishing important new methods, and we are exclusively focused on methods for basic research, so we don't publish anything in drug discovery, clinical or translational work, or diagnostic work. We tend to serve the development community in terms of who our authors are, but our readership is a broad audience of biologists. And we really emphasize conceptual advances, but we like immediate practical utility. We really like it when people can immediately pick up a new method and run with it. And one thing that sort of separates us from many of our sister journals is that we don't actually need new biological discovery to publish with us. We just need a method to show it can do something new, explore a biological problem in a new way. You don't have to have like a lengthy results or discovery. That's enough about nature methods, but I'm happy to answer more questions at the Q&A session. I just want to talk to you guys about spatial biology. So this is an area of intensely high interest for the journal. I would say the journal has supported, for example, single cell biology, next generation sequencing, all sorts of microscopy and imaging approaches since its inception. And we've had an exceptionally high interest in spatial omics technologies as they've sort of come to the forefront. And sort of evidence of this, we've published in many cross-journal collections associated with large global consortia like HCAM, HubMap, and Human Cell Atlas. And so I have images from some of the collection homepages shown here. We chose spatially resolved transcriptomics as method of the year in 2020. We had a focus issue on highly multiplex tissue imaging in 22 that I developed. And some of you may already know, we just chose spatial proteomics as the method of the year in 2024. So we think a lot of the work being done by the Human Reference Atlas and other groups is just incredibly important. It's really having a moment, and we're really excited about how far the technology has come and where it's going. So now I'm just going to move on into the specific questions, and I just put my sort of preliminary answers, but I would really love this to be not definitive in any way, but just as sort of a jumping off point for the conversation we'll have later. So this is sort of a specific question. So how can the scientific community publish an evolving human atlas that has new data elements, higher coverage and accuracy, and new use cases every 6 months? So I think that's a really interesting question, because these big atlas projects are sort of different than how science maybe has traditionally been done by individual groups, and maybe the speed at which they're done is different than like individual research projects. So I'd say my view is that there is a conventional way, which is just publish versions of the atlas that have grown or changed sufficiently to justify the next version. So let's say not only has maybe a resource grown in size, but also the way you're analyzing the data, or the way you're thinking about the data, the way you're able to visualize the data has grown and changed enough that it justifies its own publication. And I think I think this is the way people have done it. People sort of publish iterations of tools and resources. And I think a good thing about it is that obviously over time, when you do projects, the people doing the actual work grow and change. And so a new paper can easily give maybe the people who deserve first authorship or who really drove the project the ability to get their own paper and their own credit for that work. I think for funding, I can imagine that if you have more papers, it shows your productivity, and that can look good to whoever is providing your funding. And I think from a reader perspective, if you say, okay, this is the first version of the Human Reference Atlas, and now they're on the 7th version, you can sort of have a clear chronology of what each version was and how it changed, all written out in the literature. So I think those are some nice things about the way we currently do it, but you guys can argue with me. I'm not saying I'm correct, because this is just my view of it. I think some cons to this is that you might have a small change that's really important to the way you're thinking about the data, the way you can interact with the data. but it may not justify a whole new paper on its own, or let's say thinking about the project as a 2.0 or 3.0 version, a next generation of the project. And it may be difficult to convey that to the people to whom it could be important. So that might be a con. I can imagine that writing up full papers each time you have a new release or a next generation could be onerous, especially when it comes to consortia papers, because there are a lot of authors who maybe have sort of conflicting ideas about what a paper or project should be. And then I would also say, journals sort of get version fatigue, where we might have been very excited about a 1.0 or 2.0 version of an atlas, but by the time we've seen the 7th or 8th version, we're like, okay, we got it. So it might be difficult to get the attention of high-profile journals, even if the data quality, the data size, and the importance of the work is higher than ever. It could be like, oh, okay, this is the 8th version of this project. We're not as excited about it anymore. So that's a potential con. So then I think the flip side of that is maybe something a lot of journals are thinking about, which is this idea of a living publication, which is a single paper that can get regularly updated to reflect changes, for example, in large projects. And I think a great thing about this is that readers could immediately go to one document, one place, and immediately get the most up-to-date information. So maybe you're just starting to use Human Reference Atlas. You don't want to know how it was 15 years ago. You want to know how it is now. And so you could do that in one spot. I think for journals, a plus side would be if all the citations for a multi-year, multi-discipline project went to one paper. That paper would be like a blockbuster, and blockbuster papers are good for journals. Cons, I think I haven't seen many examples of this actually working or working well. So to me, it's just new. I think it's like an experiment, and I'd like to see more data before I throw my support behind it. I can imagine just like writing up a new paper for each generation of a tool could be onerous. I think constant updates could be also difficult to do. And then even with living publications, there has to be versioning and versions of records. And so I don't fully have my head around how versioning and DOI tracking could work. So I think that could be a challenge. None of these things are unsurmountable, but I just think these are maybe some of the cons. But I would love to discuss more with you guys what you think. So that's that one. So how do you best assess so this is sort of a question I thought was more both like from an editorial perspective and maybe from a peer review perspective. So how do you best assess multi-scale research that covers very different domains of expertise? For example, molecular biology, single cell biology, and pathology. So this happens a lot. So I just wanted to first say editorially, just generally speaking, how do we think about papers? And at a journal like Nature Methods. So from an editorial perspective, let us putting aside all the different types of experiments that are in a get up in paper, what are we thinking about when we read a new submission? The first thing is the topic. Does it fit within the scope of Nature Methods? For example, digital pathology or diagnostics would not. So that would immediately take a paper out of our scope. Then audience, is it of interest to a broad readership of biologists? That's a question we have front of mind. How new are the ideas and how potentially significant is the work? The greater, the better. For us at a methods journal, does the work have practical value and will it be generally applicable? Both check. Those are nice things we like to see. From then it gets a little bit more technical. Validation. Were we convinced that the method works? And then application. Did you show something cool, interesting, challenging? Something that is going to make a potential user say, aha, that demonstration resonates with me. I want to try this method. So these are the sorts of things we're looking for when we read a paper, regardless of what type of paper it is, what field it's in, or whether it's multidisciplinary. And then I consider myself the first reviewer of every paper I handle, but I can't read everything for technical correctness. We certainly rely on reviewers for their feedback in that regard. And I guess I'll talk about that a bit on the next slide. But for us at Nature Methods, our overarching question is, does it enable new biological discovery? Can you learn or do something you couldn't do before with this new method? And for us to review a paper, the answer has to be yes. So regardless of disciplines, these are the types of things we're thinking about But then I wanted to get back to sort of the specifics of that question. And the question itself reminded me of this issue I've been hearing about from many people, but it's come up a lot on social media. And it's this question of that papers become unreviewable because you have so many types of expertise that are needed. Sometimes papers have expertise that goes beyond 3 reviewers. And so what do you do in that case as an editor? Because the more reviewers you have on a study, the less likely you will publish that paper. 100% for sure, I know this. Because it really just becomes difficult to balance a lot of opinions, and you'll always find 1 or 2 people who are really sticky, maybe not mean, but really just maybe a little dogged in what they want from a paper. So if this is a real question, we always try our best, but now papers, at least in the final published form that you see, for example, in Nature, I would say we're less guilty of this than some journals. But you'll see a paper with 6 main text display items, 10 extended data items, and more than 40 supplementary items. And the burden, not only on the authors to create such a massive single body of work, but on reviewers, individual reviewers, to go through each of these things in detail and then look at the revisions. I mean, I think it's a fair question. So I don't have the answers again, but I think, at Nature Methods, we like for our papers to tell a single story. And I think that could be good for results papers too, is if you could almost pare them down and say one story you can tell with complete confidence that, and maybe break papers up a little bit more than is happening now, but just a thought. But what do we do when we have these sorts of projects where there's many different types of expertise needed? Basically, we just try to use the reviewers that have the relevant technical expertise to assess the data quality, and then give us an overall, sorry, I'm trying to move my little thing, to give us a sense of whether the main claims are supported by the data. And then when we have technical reviewers in different areas, it's our job to synthesize their advice and to take advice from diverse experts to make these decisions. And we've had a lot of experience doing this. I think Nature Methods in particular, our combined editorial expertise is something like 60 years. So we've gotten good at this, at making good team decisions. So we try our best. I will say that even though this problem seems really scary and almost overwhelming, for this particular question, many researchers are thinking across scales these days. You have very few people working in tissue biology that don't have some sense of what's going on in the single cell world. So for us, getting useful feedback is not impossible. We're able to get very good reviewer feedback for the most part. Okay, so then moving on, just in the interest of time, how can we publish 3D VR user interfaces in papers? So this is a very specific question I think Andy is very interested in. So there is no good way, to be honest. I've interacted with many VR tools, and I've never seen a way to experience even a fraction of that on a computer screen. So I don't know the answer. I'd say the conventional way for us at Nature Portfolio is just supplementary movies. That's so lame. It's so old school that I'm embarrassed that is the answer. Sorry, I have some sirens going on in the background of my house. So slightly fresher than that is that some journals can actually embed videos into the HTML version of papers. So you don't have to download anything. You can interact live. This is actually happening in Nature Portfolio. So NPJ Imaging, for example, can let you embed videos into their papers. So this is something that's getting rolled out, and I think we'll see it more. So for the VR experience in particular, I really liked what Andy showed earlier, where you can download apps to interact with data sort of separately from the paper itself. But I think if a journal or a publishing group could have that be more regular, or even have develop the tools to make that work better, I think that could be good. I don't know if anyone at our company is doing that. Then leaving that behind, I just wanted to sidestep the question of how can you share VR data in publications, but just saying, if you are a creator of VR tools, there are journals that publish these. And this is just a few examples of some we've published over the years. And I will say that they're sort of a pain to review. For the two up top, they're localization microscopy viewer, they're point cloud data viewers. So for example, for localization microscopy. And I literally had to mail around a $5,000 VR headset to the peer reviewers so they could use it. And I will say that these things still are not cheap. So this idea that it would be burdensome for some readers to have to interact with data in that way, even if it were just as a matter of clicking a link, having that technology is not fully accessible. I mean, I don't even know if you can rent it at a library or something. But that's just one thing to keep in mind. VR equipment is not, they sell like $5 ones you can put on your cell phone, but I don't think that's not good enough for interacting with the tools that you publish. You really need like the full thing, like for 3D gaming, basically. Okay, I'm sorry, I don't want to take up all day, but we have published nice tools in this space. We need our tools that we publish to go beyond data viz and allow you to look at the data in a new way to enable new discoveries. So that was a burden that each of these papers had to overcome, saying like, this is not just pretty, this is not just cool. We can do new types of analysis by interacting with the data in virtual reality. And I think each of these papers met that bar. So if you're interested in looking, please do. Okay, this one's quick. PubMap, HCA, and other paper packages have been successful in interlinking relevant papers. Could there be packages of paper packages as the Human Atlas effort grows beyond 20 consortia? Yeah, 20 consortia, that's a lot. So from a publishing perspective, publishing these huge collections is great for us because every individual paper gets a boost from being part of the collection. And so we like these collections. I think the downside is that it's a lot of work for us to do the coordination. Even coordinating two papers to try to be co-published is challenging because even if they're submitted on the same day, reviewed by the same reviewers, the revision times might be different, right? So like just coordinating on that level is hard. So I would say this sort of thing is certainly feasible, especially with a big publishing company like Nature Portfolio, because we have, like I showed you, many tiers of journals, and chances are every paper will fit in one of a journal in our family that's technically sound and passes peer review. So I think it's very feasible. It's just a matter of timing and scale. Because I think the Human Cell Atlas and the H10 collections have had 50 or more papers, but scaling beyond that might be really tricky. And I know, some papers just didn't make it. And we were like, we just have to have this in October. If it doesn't make it, it didn't make it. We just have to add it to the collection later. Okay, sorry, last question. What would publishers and editors like to publish, but nobody is submitting it? So I just took creative license with this question. So I'd say in Nature Methods, in terms of the method space, we see it all. This method space moves really rapidly. I think whenever there's hardware or software advance that is open, becomes available, people start to use it. Just like computer vision has exploded in bioimaging. Just as soon as these technologies are available, people are trying it, people are doing it. Especially anything that's conceptually straightforward. It's like, oh, that makes sense to try it. Chances are someone's trying it. And we see all of that. So I don't think there's a particular topic that I think we're not seeing that I would like to see. So maybe I would like to see the process be a little bit different. So to my view, progress tends to be like, one, cool new method, looks awesome, does good on a few demos that they show in the paper. Then it has to leave the author's hands, right? It goes out into the community, people try it with varying success, learn the pros and cons, right? There's this sort of like trial and error, painstaking dissemination phase. And then often there are many tools to do one thing. So the methods mature, and in crowded tool spaces, there are always some winners. Like maybe there's 15 tools to do a certain type of analysis, but you'll see a few, like in spatial transcriptomics, Bazor, SPA-GCN, like there's a few that for whatever reason, they're just very, very popular. And then so winners tend to emerge. And then from the winners, you sort of start to, at the end stage, see standards arising. So now when we publish these papers, we see certain sorts of standards. And I would love it if instead of this process being sort of linear and taking a lot of time and going, and a time, it's sort of a certain amount of pain, right, to get to a cool new method and then standards for publishing methods in that space. I wish there were things we could do either as publishers or as scientists to make that process smoother or maybe faster. So that's something I would be generally interested in. And then another thing I think is just, again, maybe not understudied, but I think is really important and just sort of stalled a little bit is, at least in bioimaging. So sorry, I didn't introduce myself. I handle imaging, microscopy, probes, spatial omics, certainly all of the imaging-based spatial transcriptomics, and a lot of the computational analysis tools I handle. So a thing I hear about all the time is sort of data daily, right? So, and issues with data storage, data sharing, and fair data. So I just wanted to say explicitly that I strongly support a system of federated repositories for bioimaging data with free or low-cost storage and fair data sharing. I think the United States needs to do something about this. And I know people are really working on this, and a lot of good, smart people with connections in high places, like lobbying the government, are working on this. I just want to say that it has my full support. I would love to see progress happen in that regard. And what I would love to see is whether, so even within microscopy, data sharing is just massively stalled by things like data format. What is raw data? Can you store compressed data? I mean, even that question, right, will get you 10,000 different answers. But if we came to the answer that yes, compressed data can be stored and shared, what's the optimal compression? And then the people are building better compression tools. So it's like, when do we find that sweet spot? What about file formats? What about metadata? I think I would love to see more standards, and that way the journal can be a reflection of standards that exist. And so maybe the highly multiplex imaging field has some answers to these problems. And if so, I'd like to learn them, and maybe I can use what I learned to sort of influence other areas in bioimaging. Okay, I think that's it. Yes, thank you so much. I hope I didn't go over my time too much.
It's all good. Thank you so much, Rita. That was extremely insightful. I did put your Twitter, Blue Sky, and email address into the chat so that everybody has it. Also, it is absolutely crazy that you had to ship around a VR headset to the reviewers. I can only imagine the headache that must have caused for causing delays in the review process as well.
Yes, it did. One VR headset got caught up in customs in France. And so,
so it was international too.
It was international, yeah. But as an editor, we needed that level of review, right? For software tools, we need them to be tried. So yes. Oh, I didn't say it, but 3D glasses that you can put on like in a movie theater.
Yeah.
I wonder if there's some way you could have technology like that to sort of have pseudo VR or
they are working on a different form factor, and I'm happy to discuss VR for hours. I do also want to say we have an 8 a.m. panel tomorrow morning where we talk about why 3D and why VR for multiscale human visualization. So in the audience, if you're interested in talking about this topic more, I'm going to be leading that panel, and we're going to get more into that topic. Rita, thank you so much. We're going to get back to these amazing points that you made in the Q&A part at the end. All right, it is now my pleasure to give the word over to Andrea. Andrea, do you want me to share the slides, or would you like to share the slides?
I would be delighted if you could share the slides, because that kind of takes away one very.
All right, here we go.
And Rita, can you all hear me?
Yes.
Okay. Rita, thank you very much for your nice introduction. It triggered all kinds of remarks I wanted to make. So I'm working at a data archive, or at an institution which also has data archival services. And even more so in the last couple of years, Ingrid Dillo, one of our seniors, has been extremely successful in European funded projects which talk about verification of data. So fair is fair, fair impact There's a whole set of projects. Most of them are funded in a stream related to the European Open Science Cloud, which is in itself a very interesting attempt to organize a pan-European federated infrastructure for data service providers. And I have been privileged to sit in the margins in some symposiums and discussions. And it's highly interesting, it's highly complex. Yeah, so I love what you say about it. As an archive, we also played a bit with enhanced publications we are an archive mostly for the social sciences and humanities, and our biggest collection is archaeology. We did play a bit with enhanced publications around data. And the formats are not flying, and they are, of course, there's a lot of cost involved to bring them about. So that's probably also something to touch upon in the panel, the kind of tension between cost and benefits. Kati has been with us as honorary fellow since the beginning of the 2010s, and she helped us a lot with visualization. If you can switch to the next slide. So we also promoted, and we're extremely proud to be part of the beautiful Places and Spaces exhibition, the still kind of traditional 2D maps period. And I often start my talks, but I have been already introduced with this kind of trying to make a joke out of that. And I use one of those large-scale science map visualizations. And then I say, look, I started in physics, and then I made a long journey. And then I ended up in the humanities and social sciences. Everybody who is familiar with this kind of network layout knows that it is actually a Mercator projection of a network layout, which is mimicking a globe. And so what is a long journey on the one side is a short journey on the other side. And I still like this visualization a lot because it shows that all the knowledge is somehow connected. Yeah. And that's what I find very intriguing in the Atlas project, in the Atlas of the Human Body project, is that it confronts us with the challenge that we have to go back to universalism somehow without losing everything we learned by specializing more and more and going deeper and deeper and deeper. So I think that's right on top. And the big question is, of course, can we use all these new AI techniques, all these generative AI techniques to help us to return to this kind of universal view so that we can better alternate between having overviews and drilling down and having overviews and drilling down. The problem with knowledge is, I mean, network science has contributed a lot to that, like those beautiful kind of network visualizations. And knowledge can also expressed as a network, but a network doesn't have dimensions. But as human beings, we are used to operate in something which has dimensions. We need a reference space, so we operate in 3D. So we still looking, and for many centuries, classifications were the means of kind of mimicking these orientations, these scaffolding. We still looking, and I think we need for our own brain development. We know that we have that, but also for our scientific brain development, we need to talk more about those reference spaces so that we can better understand each other. I have decided for my talk, for my remaining minutes, to actually go a bit more general and go a bit more elementary if you want, and that might not be that is kind of bringing water to the pond for all the experts in the panel and all the experts in the audience, but it probably also sheds a light on the kind of human and social communicative factor around editing and around journals. So by the way, behind that map, which you now see in the references in the slides, those points represent subfields or specialties in the sciences, which are color-coded according to a high-level disciplinary classification. And they all come from scientific journals. And the links are citations of articles in those journals among each other. If we go to the next slide, then I think I would like to remind us that if we talk about editing special issues or book collections, which I have been asked to elaborate a bit upon, they're all part of what's called formal scientific communication. Now, what the internet has given us, or the digitization and the automatization, is our incredible tools to also make our tacit knowledge, our embedded knowledge, our situated knowledge, how STS, Science and Technology Scholar, would call that, to also make traces for that. Yeah, but for the coming minutes, I will concentrate on that, what is called in technical terms formal scientific communication. And this evolved because ideas still emerge in the brain of one person. So despite of all the experiments we see, I have not seen one where somebody has connected brains, and then maybe we shouldn't wish for that. But anyhow, so it's still one brain and one person. But if it sits in one brain and one person, nothing happens. So you can be a genius in your own. If you cannot communicate that, write it down, document that so that it can be shared in one way ever, only then, if you are able to do this, then it can fly. Can be orally, can be written. And actually, if we look at journals, then it all started with the letters among scholars during the Enlightenment. And there is even a term, Republic of Letters. And if you are interested in science history, next slide, there is a long-scale research on those kind of still very known figures. And at that time, the world of in the world of the Enlightenment, at least in Europe, I think they all know each other because there were a couple of hundreds of them. Yeah, and one knows from science sociology that the human can, I think, manage about between 100 and 200 contacts. Yeah. So this slide is taken from a specific Dutch project which looked into the Republic of Letters, and then try to visualize and also to understand how the sorts between those famous groups of scholars, among which is Descartes, Huygens, Grotius, and many others, which exchanged letters. So what can we learn from the way they did it? Next slide. So the usual kind of narrative about that is that those letters got bundled in journals, and then people usually agree that the Journal des Savants is the first journal which was published in 1665. Yeah, so I think that's quite remarkable. What is remarkable is not that it has been published that early. What I find really amazing is that we still have journals, despite of the internet and despite all the other kind of forms of documentation we have now available. Next slide. So what is a journal, and what is the role of a journal in scientific communication, in what one could also call the dynamics of the sciences? Now, so the sciences have grown and differentiated into many disciplines. So those scholars from the era of Enlightenment, they worked on so many different fields. So we would call them probably universalists today. Yeah, but now we have really specialists, and we don't have disciplines alone. We have subdisciplines and fields, and in the fields we have specialties. And those structures have been investigated by a field itself, a kind of a field which analyzes the sciences, or if you want, the meta level kind of field. And already in the 50s, they found that the so-called invisible colleges are about 100 and 200 persons. I already mentioned that. And journals used to represent those cognitive structures. Yeah. So with new ideas, you have new journals. And what Rita told us before are also examples. So there comes an interest in new scientific method, or there comes an interest in specific fields or other types of genre of information we wanted to exchange, like journal types for data paper, which is a totally different beast than a scientific article, and it isn't even virtual reality or 3D. Yeah? It's just another way to writing down your data set. So whenever there are new ideas, we have new journals. Next slide. And Andreas, how I am in time? I think I should hurry up a bit.
We have 15 minutes left on the panel, so, you know, yeah.
Yeah, but we also wanted to have a discussion, right?
That's right.
So maybe I show you this because I still love the work of Lud Leidesdorff, who passed away 2 years ago and is one of the big figures in a field called bibliometrics and scientometrics, so the quantitative studies of the sciences. And Lud used to play around a lot with information coming from journal citation indices as produced by the Web of Science, and formerly by the Institute of Scientific Information, funded by Eugene Garfield. So all those kind of possibilities in the early days of automatization and digitization of our formal scholarly communication. And I depicted those two. I kind of took a snapshot from an article or an entry by an encyclopedia. Lud looked a lot into nanotechnologies, and I remember a lot of dinner kind of conversations we had with each other. And then he said, "That's really interesting, Andrea." So there was a new journal, nanotechnology, created, or several of them after a while, when the new phenomena became a bit more substantial. So it would first be reported in existing journals in this area of physics, and then people found they have enough capacity to act, and they also need a journal of their own to push for that. Yeah. And what he depicted here is that for a time, science obviously in his analysis had a lot of nanotechnology articles, and then suddenly you see nanotechnology popping up. And I think years later, or decades, or a decade, 1.5 decades later, you also see those specific journals, sometimes you see them reintegrated into existing journals. So journals show an interesting dynamics on new sorts appearing and being consumed and also taken on in mainstream, more standardized, more canonical kind of journals again. Next slide. Oh, that's the slide which should go out. That I think that's one. I forgot to delete that. So I already said that journals represent a field, and then traditionally there is an editor-in-chief together with an editorial board, and they act as guardians of their field. So they decide also what belongs to that field, what is new, what is worth to be published, and in which form it should be published. So who decides what is right or wrong, the norms they are set by groups of people. Yeah. And as Rita already pointed to, peer review is still the heart of the self-organized nature of the sciences. And I loved your example, how hard it can become to find reviewers. Yeah. For the authors, it is proof, if you publish in a journal, it is proof that you belong to a scientific community, gives you prestige, gives you also, you prove yourself as a researcher among your peers. Next slide. So that is something I copied from Kati Börner's Atlas of Science, so from the first volume of the trilogy. And you all know that. I mean, what I described you now, or what I described you so far, is almost like a family club. Yeah. So those invisible colleges, those little universities around, or these little faculties around the topic, but what then happened, what happened now, what we are still in is an explosion, yeah, of information. And how do we go about that? Next slide. Next slide. I think I have no time to go into that. So the information is increasingly also depicted as a web. And I already said at the beginning, but we humans are not spiders. So we need space. I don't know how a spider navigates in his web, but we clearly need spaces to navigate. So special issues, and that was also something I was asked, why we have special issues. They are like a topic container, and the same to a certain extent is true for book collections. And I always, I was most excited being invited into editing a special journal or a book collection, because I'm deeply convinced that we need light towers. That is something Robert Merton, a famous sociologist of science, an American sociologist of science, 50s, 60s, 70s, 80s, lots of beautiful publication. So he said, we need light towers to orient. So we can't read, even in the 50s or the 60s, they couldn't read everything which is relevant or might be relevant. So people usually orient towards those most visible, and special issues can play this kind of role. That also comes with some injustice. This is what then Merton, in his famous piece, The Matthew Effect of Science, described. Because for an individual, if you are not the Nobel Prize winner, but you had the same idea, but you were not chosen by the Nobel Committee for all kinds of reasons, most of them social of nature, yeah, where you sit in your institution, who knew you, where you're visible enough, then the credit is not given to you, yeah. But I think for us collectively, it is good that we have those light towers. It is good that we have high quality journal everybody wants to publish in, like Nature, like Science, or people know if they read something there, then it has a very authoritative high level. Next slide. And I think I'm almost to the end. Yeah, so that's a kind of, from my experience, which is limited and small, but I think the workflow hasn't changed so much. And the main ingredients is actually around a special issue or around a book collection. If you are fortunate, and I, to a large extent, I learned that from Kati by doing also things with her together. If you are fortunate, then you have your group of authors forming a group. So you building a temporary invisible college, so to say, if you do a book or a special issue. And you hope that by bringing them in closer contact, making them also reading each other's stuff, forcing, inviting, enticing people to kind of bend their contribution so that it becomes a kind of, that it becomes one piece and not really an isolated kind of collection. That's probably the best you can do. So you serve the social nature of science dynamics. And I think that's all I wanted to say. What do I have else? Those are examples. That's a bit of self-reclame. And the next one, I think, is already my yes. Thanks to the organizers. Thank you very much.
Thank you very much, Andrea, for your wonderful talk as well. We do have some time for Q&A. And so I would like to come back to something that that Rita mentioned in her talk, and there was the concept of the living publication. So the understanding, the traditional understanding is a paper is being written, it gets DOI'd, it gets versioned, and it represents a screenshot of the status of this method or this field at the time. But with things accelerating so fast, and with these Atlas projects, keeping churning out more data, the concept of something that is more adjustable becomes interesting. But Rita, you mentioned that you have not seen this kind of publication enough, or you haven't seen enough data for it. I'm curious, what kind of data are you looking for? I'm curious if Nature Methods or other journals even have a concept of a living publication, because again, right now the idea is that it's a time-stamped, DOI'd screenshot of time. So I'm curious how you conceptualize of this in your own work and what you would like to see to be more confident about it.
Yeah, that's a great question. I mean, so I actually think maybe the closest thing I've seen is the like self-versioning that people do on bioRxiv, where they might post a very preliminary version of a work and then update it and update it and update it. Or sometimes not. Sometimes they just post a version and then it gets published. But sometimes they have sort of many versions, work in progress sort of papers. But even those, typically the preprint is not the end. The end result is like a publication in the journal. So I guess, yeah, I guess I just want to see what I would be curious about in a living publication is how frequently are they actually updated, right? How frequently do the author lists change? And is there a point where it just dies? The living publication dies. You know, like, okay, they were interested in that project for 2 to 3 years. They lost their funding. The living paper died. And then it just sort of, you know, it just had its final version and that's it. And then is there any sense to the community this is concluded, or it sort of just goes on? And then I guess, how do you track? So like, could there be a functionality like track changes in Word? Like as the document grew and matured, how can you easily see what changes were made to it? Maybe if you're not a casual reader, maybe you are an interested reader and you don't want to read the whole thing again. You just want to go to what's new in this most recently changed version. Is there an easy way to do that? So I think all of these little minutia are the things that I think a company like us would want to see definitely in a trial period. So as a company, what we do whenever we have a new idea that we like is we just run a trial and we try to collect data on many, many, many dozens of papers, hundreds of papers. And then we survey the authors, we survey the reviewers. So I guess the flip side is to those questions from the author's perspective. So then the journal's job is to peer review. Peer review is part of our added value, right? So do you have to peer review every version in perpetuity? Are you obliged to continuously peer review a living document? If so, what does that look like? I think these sorts of questions become necessary, but they become very necessary because if you don't engage with peer review, then you might as well just be publishing it on your own website, right? Because then there's no quality control, there's no added value. So these are the sorts of things that if we wanted to try that, we would get all of these things sort of hammered out in a trial. And I've seen that work successfully. For example, even something as simple as double-blinded peer review, which you might think is very straightforward. We had to trial it. We had to learn from what we learned. We had to experience the trial, see what we learned from the trial, figure out the bugs, tweaks. So everything we do at the journal, we trial. But the nice thing is, we are open to lots of different formats. We want to grow and change as scholarly publishing changes, because if we don't, we'll cease to exist. So yeah, I think it'd be nice to have sort of a blueprint for that idea and then see how interested authors are. So if we started a trial and within 6 months, we only had 2 groups that wanted to engage in the trial, we would probably say, well, it's a great idea, but there's no buy-in. So it's not worth the investment of our energy. So I think that sort of thing we'd all have to get figured out.
One aspect that you mentioned that I think is really important is the temporal aspect. There's a clear timeline with, the current publication model. There's a submission, maybe upload a preprint, there's a review phase, and there's back and forth, there are revisions, then there's acceptance, and then there's publication. With a living publication, you would almost have to have a pre-established and pre-agreed upon timeline with buy-in from both the journal and from the authors. And that seems like a lot of coordination that has to be agreed, for like years in advance, potentially.
Yes, that's right. So it'd be interesting, yeah. And it's, you can imagine projects that are many decades long would have many, many reviewers over time. I've had reviewers pass away mid-review of a single paper. So you can imagine like continuity is not guaranteed on long projects. So just things to think about. I don't think any of these, as I mentioned, are insurmountable problems. It's just a matter of just outsmarting the problems, figuring them out, and B, how many groups or projects is there demand? Do people want to switch from the way they're publishing now to this sort of more fluid, more continuous, sort of long-term type of publication? I think that's a fair question also.
Do we have time for one remark? I also
Yeah.
run out. So, and Andy, I don't think you need to agree on the process. I think the beauty of the idea of a living kind of publication is, so I can imagine if you create a niche and you look for which community under which circumstances or which group would like to embrace that. Yeah, and by doing that, they also set a standard, and then you can almost observe that. So like you have your little how to say that, your little garden space where you put out some seeds and then you just see what's coming on. And Rita, you might want to look into the world of research data management plans. And people usually hate them because they are as they are right now, as they are in the majority applied, they are really documentation. So they also come with a documentation brain, which is different than a research brain. Yeah. But in essence, what some of the European funder, the European Commission also insisted on is to raise awareness that those research data management planning is iterative. And I give talks about it because we applied it in a project I put in a musicology project. I put a link into the chat. And then I see resonances from other data stewards, and they say, yes, we also see this. We need to think about research data management as an iterative process, and things are changing. We need to apply selection. Yeah, so people are often get lost in the possibilities that they can actually store or record everything, and then they don't know anymore what they finally need. Yeah. And the experiment we have been in this musicology project is actually technology-wise relatively simple. It uses GitHub, and it uses that you can read from README files in GitHub, and it produces an overview website, and it shows you data sets and their relationship to, for instance, a research question, a user persona, a tool development. So it is far less granular than all the description of the digital object, like in standards as a research object crate. Yeah, so it's far less granular, and still it is also more detailed and more interwoven into the research project than the more rigid project organization we are all familiar with. Yeah, we have our work packages, we have our tasks, we have our dependencies. And the moment you visualize where a contribution of a project consortium appears in this kind of overview, people get really interested to get their stuff in. Before we had tables, and you can take a stick. People are just tired. They don't have time to fill a table. The moment they see, oh, now my data set is one of the 26 selected by that project, important for that research question, they become interested in it. So I think by a smart combination of giving people freedom and a bit of rails, yeah, so a bit of building blocks, and then a prestigious niche. And nature, of course, is, if nature creates a niche, that's your brand power. It's prestigious, yeah. And everybody who plays in this niche is a potential standard setter for the future. So I think by this combination, and also looking into what people think with versioning in these kind of research data tracing. So I like it very much. I find the article the living art idea very inspiring. And I would say, take a self-organizing evolutionary approach. If it dies, Andy, at a certain point in time, it dies. That all, so from science, for science history, that's beautiful. We can all record that and then investigate why some die and why others go on for centuries.
And then we're going to have amazing informatics and scientometrics papers about it. This was a beautiful metaphor that you brought up. So thank you both. Apologies to the next panel. The next panel is going to be on funders. We cut a little bit into their time. Rita, I loved all of these little anecdotes you sprinkled in about reviewers. I'm sure there's many, many more. And Andrea, thank you so much for sprinkling these visualizations, bringing informatics into the mix. My PhD is in information science, so that feels right home. Thank you. I'm going to give it over to Kadi for the funders panel. Rita, Andrea, thank you so much.
Thank you,
Andy.
pleasure.
Yeah,
Bye-bye.
thank you, Rita and Andrea. I learned a lot.